The title of my research project is the effect of Aloin and Aloe-Emodin on vascular endothelial growth factor receptor in MDA-MB231 cells. Here is a quick look at my poster for this project. Breast cancer is one of the most common health problems worldwide and the second leading cause of death globally. Patients that have breast cancer have been exposed to diverse strategies towards treatment such as hormonal therapy, targeted therapy, radiation therapy, surgery, and the most common treatment, chemotherapy. The aim of such management is to improve the survival rates of patients with cancer in the earlier stages. Vascular endothelial growth factors, VEGF for short, are primary stimulants in angiogenesis that increase the vascular permeability of tumor growth and metastasis. The diseased tissue produces high concentrations of pro-angiogenic factors that further worsen the patient's condition. VEGF is known to induce endothelial cells to penetrate tumor nodules and initiate angiogenesis. Thus, as they divide, they simultaneously secrete growth factors that stimulate tumor cell growth. The vascular endothelial growth factor receptor, VEGFR2, is a highly active receptor responsible for several downstream mediators that allow cell proliferation and migration. Aloin and Aloe-Emodin both have pharmacological effects mainly through antioxidant and anti-inflammatory mechanisms. Research involving Aloin has explored its possible anti-cancer mechanisms through the downregulation of the signaling pathways responsible for the proliferation and migration of cancer cells. Similarly, Aloe-Emodin has been researched for the possible inhibition of cell proliferation and migration in cancer cells in a pleiotropic manner. Therefore, developing an anti-metastasis treatment that primarily focuses on inhibiting the interaction between VEGF and the receptor VEGFR2 with the use of Aloin and Aloe-Emodin could be a potential therapeutic option for cancer therapy. The aim of this research project is to determine the effects of aloe compounds, Aloin and Aloe-Emodin, on the migration of MDA-MB231 cells and the VEGFR2 signaling receptor. Hypothesis 1 was that Aloin will decrease cell migration at a higher concentration of treatment with or without VEGF induction, and hypothesis 2 was that Aloin would reduce VEGFR2 expression in the cells after treatment. Aloe-Emodin will slightly decrease VEGFR2 expression compared to the Aloin treatments. The compounds were acquired in solid form and dissolved in sterile DMSO at a stock concentration of 2,000 microliter. The stock concentration was then used to create the 1,000 micromolar concentration, and both treatments were stored in aliquots. The aloe concentration would reach their desired final concentration once they were added to the wells. For the scratch assay, the MDA-MB231 cells were treated in triplicates with 10 micromolar and 20 micromolar concentrations of Aloin and Aloe-Emodin respectively, and incubated for 48 hours in a CO2-free atmosphere at 37 degrees Celsius. The scratch was made using a 20 microliter pipette tip in the wells. A concentration of 50 nanogram per ml VEGF was added to the treated wells for comparison to wells without VEGF. The cell migration between the scratch was observed at the time the scratch was made and 24 hours later. For RT-qPCR, the MDA-MB231 cells were plated and treated with Aloin and Aloe-Emodin, 10 micromolar and 20 micromolar respectively, and incubated for 48 hours in a CO2-free atmosphere at 37 degrees Celsius. The required wells were stimulated with 15 nanogram per ml VEGF for 30 minutes before the RNA was extracted and the PCR procedure was followed. Figure 1.A shows the relative gene expression of VEGFR2 receptor for MDA-MB231 cells treated with Aloe-Emodin of concentrations 10 micromolar and 20 micromolar with and without VEGF. Figure 1b shows the relative gene expression of VEGFR2 receptor in the cells treated with Aloin 10 micromolar and 20 micromolar concentrations, and the VEGF-induced treatment. The MDA-MB231 cells were incubated in the treatments for 48 hours prior to the VEGF stimulation for 30 minutes in their respective wells. The extracted RNA underwent reverse transcription, and the fluorescence from the VEGFR2 products were detected and quantified using the 2 to the power of minus delta delta CQ method. To investigate the effect of Aloin and Aloe-Emodin on its possible cancer therapy potential, we assume that the relative expression of VEGFR2 will be downregulated. As indicated in the figure, there was a significant increase in the 10 micromolar Aloe-Emodin treatment in the presence of VEGF. On the other hand, there was a slight decrease in the expression of VEGFR2 in the 20 micromolar treatment induced with VEGF compared to the 20 micromolar treatment without VEGF. Both treatments had relatively higher expression of VEGF receptor in comparison to their respective control treatments. There was no significant change in the expression of VEGFR2 following pretreatment of 10 micromolar Aloin with or without VEGF stimulation. However, compared to the control treatments, there was a general trend of increasing VEGFR2 expression in the Aloin-treated cells in the presence and absence of VEGF. There was a significant increase in VEGFR2 expression in the higher Aloin treatments induced with VEGF compared to the control groups. For the scratch assay with Aloin-treated cells, there was a significant decrease in cell migration of Aloin-treated cells in the presence of VEGF compared to their respective wells without VEGF present. The lower concentration treatment, 10 micromolar Aloin, had a greater cell proliferation than the higher Aloin concentration in the absence of VEGF. However, the deviation between cell migration in 20 micromolar treatments is within the range of the average cell migration values of the 10 micromolar treatment. The vehicle control group had a treatment of 1% DMSO and PBS plus 5% trehalose to indicate the effect of Aloin and VEGF on cell proliferation without the interference of the solvents present in the treatment. The Aloin treatments 10 micromolar and 20 micromolar induced with VEGF both exhibited a similar amount of cell migration within the scratch width, although the 20 micromolar treatment had more deviation between the biological replicates. For the scratch assay with Aloe-Emodin treated cells, the data indicated that there was a trend of decreasing cell migration in treated cells compared to their respective control groups. As the concentration of Aloe-Emodin increased, there was a slight decrease in cell migration. Furthermore, the migration decreased in treatments with VEGF present. There was a high amount of deviation in cell migration in the 10 micromolar Aloe-Emodin with VEGF absent. Additionally, the cell migration is lowest in the 10 micromolar treatment induced with VEGF. The data indicated that there was a general trend of decreased cell migration of pre-treated MDA-MB231 cells in the presence of VEGF. Compared to the data of their respective group without VEGF, there is a great difference in the cell migration. Metastasis of the tumor cells from one location to the other parts of the body is a main indicator of the progression of breast cancer. Proangiogenic factors such as VEGF play an important role in the migration of breast cancer cells and promote tumor progression and metastasis. The regulation of VEGF receptor 2 and its role in cancer development is largely unknown. However, targeting the binding of VEGF to its receptor with the objective of blocking the receptor with another compound could prove a novel mechanism for cancer therapy. Due to the harsh side effects patients face from existing breast cancer treatment, there is increasing focus directed to cancer therapy derived from naturally occurring compounds capable of treating tumor growth. In this study, we have proposed the use of allo compounds, Aloin and Aloe-Emodin, as possible inhibitors of cell migration in MDA-MB231 cells. By inhibiting the interaction between the proangiogenic factor, VEGF and its receptor VEGFR2, there is a possibility in reducing the cancer cell to endothelial cell communication that is involved in tumor metastasis. The treatment of Aloin led to a decreased cell migration in MDA-MB231 cells in the presence of VEGF. Interestingly, the expression of VEGFR2 in Aloin-treated cells had increased significantly in the presence of VEGF, as shown in Figure 2. This occurrence does not support the proposed correlation between the binding of VEGF to the receptor and its relationship with cell migration. It was hypothesized that blocking the binding of VEGF and VEGFR2 would ultimately decrease the tumor cell migration. There was a great amount of deviation between the cell migration in the wound healing assay. This may be due to the rinsing of detached cells and addition of fresh media to the wells. These actions may have caused the discrepancies in the control wells. Due to the agitation from the addition of fresh media following the scratch formation, the cells may have detached and settled on the scratch rather than migrate from the surface of the well. Interestingly, the 10 micromolar Aloe-Emodin treatment induced with VEGF had a lower cell migration compared to the control group and the higher treatment concentration. Yet there was an increase in the relative expression of VEGFR2 of MDA-MB231 cells treated with 10 micromolar Aloe-Emodin in the presence of VEGF. This was similar to the effect of Aloin on decreasing cell migration, yet increasing the expression of VEGFR2. Investigating a broader range of treatment concentrations of Aloe-Emodin with longer incubation periods could potentially decrease cell migration. We can assume that the Aloe compounds may prove effective at decreasing the cell migration of tumor cells, yet this is not accomplished through the downregulation of VEGFR2 in MDA-MB231 cells. Another possibility for the decreased cell migration and increased VEGFR2 expression in the treated cells would be due to hypoxia stress. Hypoxia occurs when the cells have been deprived of oxygen due to the rapid increase in cell growth. In response to the hypoxic stress, the cells are able to undergo changes in gene expression to facilitate the growth of the cells. Some studies have shown that VEGF and its receptors were significantly upregulated in response to hypoxia. The increased confluency of the MDA-MB231 cells would have resulted in the occurrence of hypoxia in the cells during the treatment incubation period, leading to the upregulation of VEGFR2 expression. The findings suggest that Aloe-Emodin is a potentially therapeutic option for cancer therapy that targets the VEGFR2 receptor, in turn decreasing the effect of VEGF on the proliferation of the MDA-MB231 breast cells. However, the decreased relative expression of VEGFR2 through Aloe-Emodin was not significant in this study. Therefore, a wider range of treatment concentrations with a large number of biological replicates is required to determine the reproducibility and downregulation of VEGFR2 expression in MDA-MB231 cells. Both treatments of Aloin and Aloe-Emodin had an effect on the tumor cell migration of MDA-MB231. However, it was not through the blocking of the VEGF receptor. Thank you for listening. I would like to thank Dr. Michael Gibson for his expertise and advice throughout the academic year. I would also like to thank Dr. Jessica Livingston for her organization and guiding presence throughout the process of this research project. I would also like to thank Crystal McFadden and Marla McKenzie for their knowledge, assistance, and overall great company when I needed it. Thank you so much.